Hello, my name's Inez Januszczak, so I'm from the Natural History Museum, and obviously I know a lot of people in this room already regarding the Darwin Tree of Life project, which I will obviously mention at the end, but I want to introduce another insect-related genomics project called Polygen. So I'll be just doing an overview of that, as well as sort of highlighting some of the ways you can get involved, and also explaining some of the genomics behind it as well in hopefully a simple and clear way. So as mentioned, I am the sampling coordinator for the Darwin Tree of Life project. I'm still the sampling coordinator for the Darwin Tree of Life project at the Natural History Museum, just technically 1 day a week, but I'm still involved. The aim for the Darwin Tree of Life project was to sequence the whole genomes of every eukaryotic species in the British Isles, approximately 77,000 species, but this includes all plants, fungi, animals, including invertebrates, chordates, etc. So at the Natural History Museum, the focus was mostly terrestrial, freshwater and marine invertebrates, as well as vertebrates. So just a caveat that I'm not a Diptera expert, but obviously I've worked with a lot of different Diptera in terms of getting them through the pipeline and some of the other groups, and just highlighting a little bit of the process. So for the Darwin Tree of Life, we had to flash freeze identified specimens, which were then DNA barcoded and then dissected, then sent to the Wellcome Sanger Institute in Cambridge, so here, where they did the human genome for the first time, just highlighting one of the genomes that has been recently released, which is the Melanomia nana, and I just thought I'd highlight some of the people who are involved in that genome, whether it is collecting the specimen, note writing, or providing additional material. And I think all 3 of the people are actually in this room today, so that genome was released very recently. But in terms of Polygen, it is a slightly different angle in that we are also working with museum specimens. Erica, I stole this from your Facebook post from like 6 years ago. This is obviously a tray of Joseph Banks specimens from the museum of flies. We're not working with stuff this old, but just to highlight, obviously in the museum, we have a huge collection of Diptera. So for Polygen, this NERC-funded project, which is investigating genome diversity change in the UK, and it's in collaboration with the UK Pollinator Monitoring Scheme, Bumblebee Conservation and Natural England. And this is using both museum and modern samples. So the timelines are quite specific. So for modern, it's anything collected within the last 2 years or now, and museum samples looking at subsets from the 1930s, 50s and 70s. And it was quite intentional with the time periods that were chosen, which I'll explain in a minute. And the project is running from May this year, 2025, to October 2027. So by the time we did manage to have the species list in place, etc., it was a bit further along in the year, but hopefully next year is when we'll be able to really carry out the majority of the sampling effort. So why pollinators? I'm assuming that this is a quite good audience for this, but essentially in the UK, there are around over 1,500 wild pollinators listed. And as we all know, pollinators are facing a huge decline, despite the fact that they are responsible for, and this is just, I'm quoting one paper from Simon Potts, which is they're responsible for 75% of pollinating crop species, 35% of global crop production, and 88% of flowering plants. So, you know, obviously a huge, kind of a huge member in terms of contributing to all these kinds of ecosystem services. And I'm also quoting a paper from Powney in 2019, strategically chosen because it involves data both from the British bees, wasps and ants recording scheme and the hoverfly recording scheme, in that 33% of wild pollinators have declined between 1980 and 2013. And I've just mentioned that less known is the role of flies in the pollinating cycle. Not here, but I think in the UK generally, not as many people realise the role that flies have in pollination. And I just thought this infographic was very helpful from the Powney paper, which shows the difference between the hoverflies, which is orange, and then the blue is the bees. It's one of the few papers I found that does actually distinguish between the two and shows this decline is visible in not just bees, but across all pollinators in general. So why museum samples? With museums, the advantage is there is a historical time series. Ideally, I write ideally because actually a lot of museum collections are quite biased. And you do find, or I've definitely found, that the beautiful time series you would expect from a museum collection isn't always the case due to things like who's interested in what species at what time, the funding available, the connections available, etc. Secondly, there ideally would be a diverse geographical spread with museum species. Again, ideally, I say this because, again, having looked at the museum samples, it's a bit biased across the UK. So Natural History Museum definitely has a southern bias when it comes to UK collections. However, only a leg is required for the genomic work that I'm working on at the moment. So at least it means that you can retain most of the sample and use a subset of it for any kind of genomic research. So the requirements for species selection for the Polygen project. So there was quite a specific subset of criteria that was written up in the grant itself. So the first one that was that a whole published genome must be available. So as mentioned through the Darwin Tree of Life project, the aim was to publish the whole genome of a specific species. Not all the species have been done, so we had to select the subset of species that have a whole genome available. Secondly was that the genome was easy to analyze. There's been a huge number of fly genomes published, but not all of them were easy to analyze in the sense of a lot of junk DNA, a lot of repetitive DNA, too big, too small. Luckily, hoverflies were quite a good contender. As I was told, they were very small genomes, I think only a few chromosomes as well. So they were a very good group to focus on initially. They need to have a well understood ecology and be of vague ecological interest, so to fit with the pollination theme, easy to identify and collect, and have strong recording data. And as mentioned, luckily in the UK, we have lots of recording schemes that are extremely active. So we do have a lot of data for a lot of these species. And obviously the final one is available in museum collections, but with the caveats that I just mentioned, in that perhaps they're not as, the samples may not be necessarily as available as we hoped. But that sort of became more evident as the project continued. Just to mention that all the genomes are available online. This is just a QR code for Episurface Voltiatus, but the ones that I will mention are also available online. So why genomics? So I was hoping to just vaguely cover the different methods and techniques used, since I've now mentioned 2 different genomics projects. So just to clarify what the purpose of each one. So as mentioned, the Darwin Tree of Life project was the whole genome. So this is an organism's complete set of DNA. Already, I think Denise has mentioned DNA barcoding. So this is a short, specific region of DNA. And some of the projects involved with this are the UK Barcode of Life, or indeed BioScan. You may have also heard of genome skimming. So this is sequencing sections of the entire genome at low coverage, which essentially means it is the whole genome, but only sequenced a few times. And this was mostly used for Biodiversity Genomics Europe, which is another project that is looking at museum specimens. So there's various projects working with various different types of sequencing techniques, again, to result in various different types of data. So I placed Polygen around here. So for Polygen, the aim is for 12x coverage, which means every section of the genome is going to be sequenced at least 12 times. So not as much as whole genome, where each section is sequenced about 30 to 50 times, but similarly not as little as genome skimming. This sweet spot of genomic coverage means that we are able to do a lot more in-depth analysis on the genome that we can't do with skimming because it's not as in-depth. However, we need a whole genome to be able to do the comparative analysis. Hence, this project is only possible because of the work that the Darwin Tree of Life project did previously. So by using whole genome and short read data, this means we can study the museum specimens where the DNA may be more fragmented and do it more cheaply. So publishing a whole genome is extremely expensive. Whilst skimming is obviously cheaper, by increasing the amount of coverage, this means we can do a larger variety of evolutionary related research. For example, looking at the population structure, genome diversity, ecological correlation, and signals of selection. So I was really happy to see actually Denise's talk about the effects of climate and how populations have moved across the UK. The dream is we have that data combine it with then the genomic data, and that way we can actually track evolutionary change as these populations move across the UK. So I mentioned a few of these species already. So these were the species that have been selected for the Polygen project in terms of Diptera. So as noted, they all fit the criteria I mentioned before. So they have a whole genome released. They're easy to identify. They are good records. There are some caveats in that. So Ceratipipiens was submitted to match the EU pollinator monitoring scheme project. And the Leucozona laternaria was a specific request by Roger Morris to include it in this. However, the only person I've met who's actually seen this has been Jane Hewitt, and no one else has said they've seen it. So I actually think it's quite rare. However, we are including it because there are enough specimens in the collection. So it would be brilliant if we could get enough modern material for this species. And just to also highlight the other species on the list. So these are all the bumblebees and wasps. So this is the total species list now for Polygen. And as you can see, it's just a good range of very common bumblebee, solitary bee and wasp species that you can find across the UK. So how to get involved, and this is just one of the reasons why I'm here to promote this project, is because if you would like to get involved, we would like modern material from across the UK, and we would want as high as geographical spread as possible. So the criteria is for the specimen to be collected in 70% ethanol, which we would provide. We need a different site per species, ideally not within a 16-kilometre or 10-mile radius. So the idea would be to not collect the same species, multiples of the same species on the same site, but across the UK. Of course, permission granted, but if you are near a garden or a public path where you've seen some of these species before, that would be ideal. The specimens need to be fully labelled with all the relevant collection data and then sent to the museum, and we can provide stamps and envelopes as well. So do find me in the break if this is of interest and you would like to be involved in submitting hoverfly or indeed other the bees and wasp specimens for this project. And I'm more than happy to talk to you about that process. So yeah, that's Polygen. I also thought it would be good to shoehorn in a Darwin Tree of Life update. So as mentioned, this was to sequence all the whole genomes of all the specimens in the animals in the UK. But the Dipteras Forum has played a huge part in this in the last 4 years I've been involved on the project. So as just a very, very small highlight, through the Darwin Tree of Life project, around 407 Diptera genomes have been assembled. And out of these, 124 have gone through the NHM. So this is a huge chunk. And like I said, most of these have been submitted by members of the Dipteras Forum. So I've done a bar chart of the families that have been sequenced so far. Unsurprisingly, Serphids and Tachinids are leading the way, but we still managed to do 38 families, which is absolutely brilliant. So for this 38 families, 46 collectors, and you just sort of also here to say a huge thank you to the forum, who's been extremely supportive in submitting specimens to the Darwin Tree of Life. And we are still accepting specimens. And so if you do think you've got some of these less popular species around there where you live and they'll survive a postal journey to the NHM, do let me know. And of course, if you want to track some of the other species available, then you can find these on the Darwin Tree of Life portal. So yeah, thank you very much to everybody here. Again, if you'd like to submit specimens for Polygen, do let me know, or if you want any more information about anything else. And I would just like to also say a huge thank you to all the Diptera curators at the Natural History Museum, and of course, all of you who have submitted to all specimens to either project so far. Thank you.
Have any questions?
Yes.
Can I just make a point about Leucozona?
Yes, Roger wants to make a point about Leucozona laternaria. Yeah.
If I remember rightly, you had a series of caveats as to what you wanted, and that was why that was suggested.
Yep.
The other three, or whatever, are highly cosmopolitan.
Yes.
And to a very large extent may be affected by migration.
Yes.
So it was inevitable that you wouldn't see it in the numbers that you would Epicervus boltiatus.
Yep.
But it is pretty widespread.
Yep.
And it is one that you will come across if you are a serious dipterist. So
Yep. Noted.
How? How everyone.
simply a question of the type of sampling that you participate in. Everyone can pick up episurface Borthiatus,
Yeah.
but only a proportion of people will be looking in such a way. So you're going to get different information out of those. I would add a further point. The Myothropa, it's worth keeping a very careful eye on it because there are suspicions in Europe there are actually 2 species in Myothropa. So
Absolutely.
I would be very interested. I think Sharon Van Steenis would be extremely interested in the data.
Absolutely. Yeah. So Roger's point was, shall I repeat that or?
Yeah, go.
Okay, no, Roger's point was if you are a serious dipterist, you'll have no issues. And also that obviously some of these are migratory. We know they're migratory. That actually is not an issue because, like mentioned, we are looking at the evolutionary change of the species. If we are looking at the specimens from the museum collected in the UK in the 1930s, it is still an interesting research point to see how much they've changed, how much migration has there been between those 2 different time periods. And I think, we've had a lot of comments about migratory, populations moving, etc. All of that is going to be feeding into the data. And the key is because we can do this comparison to around 100 years ago, that is what will be interesting to see. And I think that even no one's actually tested the levels of migration or how much the speciation has happened, etc. So hopefully those will be some of the things that we can answer through looking at this data. Yes, Stephen, yes. Yeah.
Am I right in thinking for Darwin Tree of Life, you still need to get a live specimen to you rather than a dead one?
Correct. So Darwin Tree of Life, because it's whole genome and requires to be frozen to get the amount of genomic data out, it's still live specimens for Darwin Tree of Life. For Polygen, like I said, it's just in ethanol is fine, and even room temperature as well.
Any questions online? No. Thank you very much.
Thank you very much.